Namaste, I am JVN. I am Dr. Anupama GOYAL. I am today delivering my lecture on sphagnum and I am working as professor in Jyoti Vidyapeeth Women's University, Jaipur. So students, this lecture on sphagnum will be beneficial for the students studying in BSC as well as in MSc classes. Well, first I talk about the systematic position of sphagnum. So it belongs to division Bryophyta, class is Bryopsida and subclass is Sphagnobria, order is Sphagnales and family is Sphagnaceae. Now I talk about the occurrence of sphagnum. Well, the genus Sphagnum is commonly known as bog moss or peat moss, and this sphagnum, which is bryophyte, is cosmopolitan in distribution, having around 340 species, and the species of sphagnum are aquatic or semi-aquatic plants and grow in swamps, ponds and moist habitats such as along the banks of lakes, wet hillsides, and the species of sphagnum are perennial. They produce certain acidic substances as a result of which the aquatic habitats in which the sphagnum grows becomes unsuitable for the growth of fungi, bacteria and other pathogens. This slows down the decaying process of the dead parts of the plants. Well, the gametophytic tissue of these perennial plants of sphagnum after becoming dead settles down along with other partly decomposed dead organic matter at the bottom of the water body. This partially decomposed dead organic matter gets compressed over a period of time by their own increasing weight and gives rise to a hard black colored substance which is called as peat. In the long course of time, this peat gets carbonized and is used as direct fuel of great economic importance. Now I will talk about the structure of sphagnum. Well, first of all, see the external features of the bryophyte sphagnum. The gametophytic phase of sphagnum is represented by two distinct stages, namely juvenile protonema and mature leafy or gametophore stage. Now the mature plants of sphagnum grow in dense clumps and their shoots are of whitish or brownish green in color. All the species of sphagnum accumulate water and grow with bright color, that is deep red, rose pink, and due to the presence of water soluble pigments called as anthocyanin, they are perennial, showing unlimited growth by means of an apical cell with three cutting faces. Very young gametophytes bear multicellular rhizoids with oblique septa. Mature gametophytes do not bear rhizoids. It is differentiated into an upright branched axis and leaves. So students, what is the function of rhizoids? Rhizoids help in encouraging the plant to the soil substratum, and they also help in the absorption of water and mineral nutrients from the soil. Now this picture shows the adult leafy gametophore of sphagnum. It shows the pseudopodium, the capsule region, and now I talk about the internal structure of sphagnum. First of all, the main axis. So the main axis of sphagnum is differentiated into three distinct regions. First is the cortex. So now I talk about the cortex. The cortex forms the outer part of the main axis. In the main axis, cortex is made up of 2 to 4 layers of thin-walled living hyaline cells, but in the axillary branches, cortex is single-layered. The mature cortical cells lack protoplast, and the cortex of the main axis is single-layered in sphagnum subsecundum. In the lateral branches of sphagnum molluscum and sphagnum tenellum, the cortical cells elongate and become bottle-shaped or retort-shaped. The neck of these cells is curved and has a pore at the distal end. These cells are called the retort cells, and they accumulate water. So what is the function of the retort cells? They help in the accumulation of water in sphagnum. Now the prosenkymatous region. So this region lies inner to the cortex, and it is composed of 4 to 6 layers of small, thick-walled and narrow parenchymatous cells. The prosenkyma provides mechanical support to the axis. This region is also called as hydrome. So another name for prosenkyma is hydrome, which helps in providing mechanical support to the axis of sphagnum. And now the central cylinder, the innermost region of the axis, is called as central cylinder or medulla. It is made up of vertically elongated, thin-walled parenchymatous cells. So here in this diagram, it is showing the TS of the old axis in sphagnum. Outermost part, you can see the cortex. Then inner to the cortex, there is the prosenkymatous region. And then you can see the central cylinder in this diagram. And now I talk about the, I've already told leaf. Leaf in sphagnum, a cross section of a young leaf of sphagnum, shows rectangular or square cells of uniform size. The mature leaf has the following two types of cells: chlorophyllous cells and the hyaline cells. So the chlorophyllous cells are small living cells with many discoid chloroplasts in sphagnum acutefolium. In sphagnum acutefolium, the cells are triangular. In sphagnum squarosum, the cells are biconvex, and they are also called as photosynthetic cells due to their photosynthetic ability. Now, what are the hyaline cells? These hyaline cells are large, polygonal and colorless cells. These cells lack protoplast. The walls of these hyaline cells have spiral thickenings and pores. These cells are also called as capillary cells, as these cells absorb water. The chlorophyllous and hyaline cells are arranged in an alternate sequence. So this section shows the transverse section of leaf of sphagnum acutefolium. Well, A section shows the sphagnum acutefolium. B part shows the sphagnum tenellum. So this is the leaf of sphagnum tenellum. And C, this part, it shows the leaf of sphagnum squarosum. So here in this diagram, you are seeing the transverse section of the leaf of sphagnum acutefolium in A diagram, in B diagram sphagnum tenellum, and in C diagram sphagnum squarosum. Now the reproduction, I talk about the vegetative reproduction in sphagnum. Well, it takes place by the following methods. First is innovation, Second is multiplication of protonemal branches. Now, what is innovation? It is the common method of reproduction in sphagnum. It takes place by the formation of special vegetative branches called as innovations. Occasionally, one of these branches in the axillary cluster become robust and grows upwards. This branch shows all the characteristics of main axis and is known as innovation. Each innovation develops into a new plant when it gets detached from the parent plant. Now, the multiplication of protonemal branches, any marginal cell of the primary protonema may become meristematic and forms a green cellular filament in apical portion, grows into a flat thallus-like green secondary protonema. And now I talk about the vegetative reproduction in sphagnum by regeneration, the growth of new tissues or organs to replace those lost or damaged in injury is called as regeneration. So, sphagnum has great power of regeneration. During desiccation, the growth of sphagnum is checked because the physiological activities like respiration and photosynthesis are suspended. What I mean to say is, when the conditions are not favorable or when the during desiccation is there, then the activities, physiological activities like respiration and photosynthesis are not carried out by sphagnum, but the cytoplasm shows a high degree of resistance to desiccation. When water is available, these activities like respiration and photosynthesis are resumed and normal growth of the plant takes place. Such plants are known as paleocophytes. Now, how the sexual reproduction takes place in sphagnum. So, first of all, I will talk about the development of antheridium in sphagnum. Well, the antheridial initial grows into a papillate outgrowth, then it undergoes transverse division to form a small filamentous structure. The terminal cell of the filament undergoes two oblique intersecting divisions, resulting in the differentiation of an apical cell. The apical cell has two cutting faces and forms derivatives in both the planes. This results in the formation of a 12 to 15 cell structure. 2 to 5 distal cells form the body of the antheridium and the remaining cells form the stalk. The distal cells undergo periclinal divisions and each gives rise to an outer jacket initial and an inner primary endogonal cell. The endogonal cells divide in all possible planes. The last generation of these cells forms the endocytes. The jacket initials undergo division to form a single layered jacket of the antheridium. So here you can see A to G shows the successive stages in the development of antheridium in sphagnum. Here in the A diagram you see the development of antheridium starts from the antheridial initial, and B diagram there is the formation of the apical cell, then there is the formation of the primary endogonal cells in D diagram, and so A to G stages shows the successive stages in the development of antheridium. Now the mature antheridium of sphagnum. Well, the mature antheridium of sphagnum has a globular body which is borne on a long stalk. Stalk is made up of 2 to 4 rows of cells. The body is surrounded by a single layered jacket of sterile cells. Now the dehiscence of Antherium and sphagnum. When Antherium matures, the apical cells of the antherial jacket swell by absorbing water. So due to the turgor pressure, antherial wall breaks into a number of irregular lobes at the apex. These lobes turn backward, which exposes the mass of endocytes. When the endocytes are liberated from the antherium, anthrozoids come out. And now I talk about the development of archegonium in sphagnum. Well, the archegonia develop on the apex of the archegonial branches, either singly or in groups. The apical cell of this branch forms the primary archegonium. 2 to 5 secondary archegonia develop from the derivatives of the apical cell. There are 3 archegonia in a group, that is, one primary archegonium is at the apex and 2 secondary archegonia emerge from the base of the primary archegonium. The archegonial initial divides transversely to form a 4 to 6 celled filament. Then the terminal cell of the filament divides by 3 intersecting vertical walls, resulting in the formation of 3 peripheral jacket initials and a primary axial cell. The primary axial cell divides transversely to form an upper cover initial and a lower central cell. The cover cell divides vertically, forming a group of 8 or more cover cells. These cells form the jacket of the terminal part of the archegonial neck. The central cell once again divides transversely and gives rise to an upper primary neck canal cell and a lower primary venter cell. Now the primary neck canal cell undergoes repeated transverse divisions to form a row of 8 to 10 neck canal cells. The primary venter cell divides once transversely to form a venter canal cell and an egg. At the same time, each of the 3 jacket initials undergoes anticlinal and periclinal divisions, forming the middle and basal portion of the archegonial jacket. Now repeated transverse divisions of the jacket cells result in the formation of 6 or 5 vertical rows of neck cells. So this diagram shows the successive stages in the development of the archegonium and sphagnum. See, A diagram shows the primary archegonium cell, and now from the primary archegonium cell, secondary archegonium initial is formed, and in the last you can see the mature archegonium having the cover cells, primary venter cell, venter canal cell, and the egg. Now I talk about the sporophyte in sphagnum. Well, the diploid zygote is the first cell of the sporophytic generation. Among the few archegonia, only one is developed to form embryo in an archegonial branch. The zygote enlarges, it invests itself with a cell wall, and then the zygote divides transversely to form an upper epibasal cell and a lower hypobasal cell. Now the transverse divisions in both the cells continue until a 6 or 7 celled filament is formed. The lower half of the filament undergoes irregular divisions, forming a parenchymatous bulbous foot. The foot acts as a haustorium. The upper cells of the filament divide by two vertical divisions at right angles to each other, and a quadrant is formed. The cells of the quadrant divide periclinally to form an inner endothecium and an outer amphithecium. Now, the cells of the endothecium divide repeatedly and form a central sterile part called as the columella. The amphithecium divides periclinally to differentiate into an inner 2 to 4 layered archaeosporium and the outer 3 to 7 layered capsule wall. The archaeosporium forms a dome-shaped arc over the columella. The cells of the archaeosporium later develop into 2 to 4 layered sporogenous tissue. All sporogenous cells function as spore mother cells that divide meiotically and form haploid spores. The spores are enclosed within a spore sac developed from the surrounding sterile tissue. There is only a short neck-like inconspicuous seta connecting the upper capsule and the lower bulbous foot. Now the structure of the mature sporophyte in sphagnum. Well, the mature sporophyte in sphagnum consists of a bulbous foot, a neck-like inconspicuous seta, and an almost spherical black to dark brown capsule. The whole sporophyte is covered by calyptra, and the lowest part of the calyptra that covers the foot is called the vasinula. The pericardial leaves are present below the sporophyte, and the elongated archegonial branch at the apex of the sporogonium is called the pseudopodium. It increases in length and pushes out the capsule above the pericardial leaves to facilitate the spreading of spores. The capsule in longitudinal section shows an outer zacket and middle spore sac with spores. Now, structure affinities of sphagnum. Well, sphagnum shows resemblance with liverworts like Hepaticopsida, hornworts, Anthoceratopsida, and mosses. It possesses certain characters in which it stands apart from all the three. These are, first of all, resemblance with Hepaticopsida. First of all, thyroid protonema resemble the juvenile stage of some acrogynous jungermaniales. Second, position, structure, and dehiscence of Anthridium is like jungermaniales, for example, Porella, and position, origin, and development of Archegonium is similar to that of acrogynous jungermaniales, and these resemblances between the liverworts and sphagnum point to its intermediate position between the liverworts and mosses. So all these resemblances between the liverworts and sphagnum point to its intermediate position between the liverworts and mosses. Thank you.